Good morning, everyone. Today we're going to be talking about antibiotic resistance in bacteria. And so the slideshow that I'm going to be going over is the one that I posted for you this morning. If you want to open up and follow along, that would be great. So today we're going to be talking about microbial connections and the rise of antibiotic resistance. We're going to be learning about this both in honors biology and in rise biology as kind of a case study in natural selection and evolution. So the first thing to talk about today is really just the outline of activities. And then I'd like to go back to the title slide and discuss that quite a bit. So first of all, this presentation really has three main parts. First of all, we're going to be going over a brief introduction to what antibiotics are and what it means to be resistant to antibiotics in terms of those bacteria. Second, we're going to cover why antibiotic resistance is such a large health concern and how a world with no functional antibiotics would be a very unpleasant place to live in. And then third, we will cover what is currently being done to combat the antibiotic resistant crisis, followed by, I'd like to talk to you a little bit about our simulation that will be introduced as soon as this presentation is done. And that's going to be a game that hopefully you'll enjoy playing with your family if you'd like to, or at least collecting the data and watching the simulation that we've made for you. But it's a marshmallow and M&M, like little mini lab game that you can play just to show how antibiotic resistance comes into play with bacteria. So going back to the first slide, this is actually a slide depicting a plate that's called a blood agar plate. And so what you're looking at actually contains mammalian blood. And usually this is done using sheep or horse blood or something of the sort. But it actually shows us hemolytic activity. These are some of the bacterial colonies you can see in the middle of the plate. And if you notice around them, there's almost these halos that are created. And what that means is that they're lysing the red blood cells that are on the plate. And as that lysis happens, of course, the hemolysis, okay, hemo meaning blood, lysis, everyone knows that term at this point, means to burst or to break, right, to split open, has caused all the hemoglobin inside the red blood cells to leak out, which is why you no longer see the red, okay, in that zone that is depicted as kind of the halos there. So what that means is that some of these bacteria that are being grown on this plate, and this is a little bit scary, actually produce what's called exoenzymes, and those are able to lyse the red blood cells and actually also degrade the hemoglobins. And so again, this is an agar plate. Most of us this year have seen these before. We've done some bacteria experiments. So again, I hope all this is interesting for you as we get into bacteria and their ability to, unfortunately, resist our antibiotics. So moving along, let's go ahead and start talking about bacterial warfare here. And so the idea is that bacteria live in these really harsh environments that are very often nutrient poor, and we could say they must compete with each other then for these limited nutrients. And so another thing to think about here is the really length of time bacteria have been around. Most of you guys remember how old the Earth is. We had discussed that it's about 4.6 billion years old, right? It's estimated to be very, very old, billions of years. Not millions of years like some of you guys thought at the beginning of this entire unit. Well, it's estimated that bacteria have been around a very long time as well. They've been around for about 3.5 billion years, it's estimated. And evidence really suggests that they've been present since early Precambrian time. So kind of keep that in mind throughout this talk today. In essence, what that means is bacteria, because they've been around so long, have really been able to compete with each other. And over billions of years, they've evolved the ability to harm other bacteria in order to increase their odds of surviving. So secreting chemical toxins is one way that bacteria can actually protect themselves from other bacteria. And it's kind of like a fighting response that they start coming up with these things to kill each other off and protect themselves. So the introduction of antibiotics as chemical toxins, right, was actually catalyzed by the idea that maybe humans could capture these chemical toxins and use them against harmful bacteria that are found in us and other mammals. And so the scientists that worked on the first antibiotic, if you've heard of Fleming before, I'll discuss him in a minute, were very smart, and they were quoted as predicting that harmful bacteria then would probably quickly evolve to combat any antibiotic that humans began using in large quantities. So in other words, they foresaw the problem that we're going to be talking more in detail about today. So don't forget, Antibiotics, what these are, is really just these toxins, the same toxins bacteria produce in mass quantities to kill those own bacteria that harm mammalian cells. And so antibiotics are those toxins that we now can produce that kill bacteria. And so they can kill bacteria in a variety of ways, as I'll mention shortly, by using different cellular targets to target different parts of those prokaryotic cells that we learned about first semester. So if you've heard of Alexander Fleming, he, of course, is credited with the discovery of antibiotics. He discovered the very first antibiotic when he was studying the bread mold, I believe it was penicillium. And so he actually noticed that the fungus penicillium killed the disease-causing bacteria on the plate. And this is all very accidental, guys. This is all very much by accident. It was totally unintentional when this was discovered. But Fleming did realize the potential for penicillin after he witnessed this fungus kill the pathogenic bacteria on his agar plates. And so, how does this actually look? I wanted to talk about this a bit. Antibiotics that target bacteria are actually shown here on these kind of clearish discs that you see. They're actually whitish discs, I apologize. And the clear zones around them, those are called zones of inhibition, and that's given down here at the bottom. And so notice that the antibiotics would actually be soaked into these little white discs, pre-lab or before you did this, and then they're placed on the plate, and then the bacteria, of course, is also placed on the plate and allowed to grow for a certain amount of time. So the bacteria here is depicted by what you see. It looks like a lawn. Okay, it's all this kind of like yellowish lawn-like cells here that are all growing together. And so that's bacterial growth. But notice around these small discs, we have this clear space. And some of the clear spaces have larger diameters, like this one here I was just pointing to, than others. Okay, there's a smaller diameter of clear here and a very tiny diameter here, almost none at all, right? And so sometimes what we're going to see is that this can show us how effective antibiotics are because of the fact that some antibiotics are very good at killing off bacteria. It depends on the type we're growing on the plate too, of course, whereas others, like these over here, really don't seem to be killing off very many bacteria at all. So depending on that type of bacteria we're growing on the plate, the antibiotic may or may not be effective in killing those bacteria, but zones of inhibition are how we can actually measure how effective an antibiotic is. So remember that those discs have already been pre-soaked in certain types of antibiotics, and each one could have been soaked in a different type of antibiotic. So we're actually looking at, if this is all one type of bacteria, how effective these different antibiotics are for killing off a certain type of bacteria. All right, and so this is how we can actually measure that and look for resistance, right? If some of these bacteria aren't being killed off, obviously the reason is because they probably, as we'll learn soon, have resistance genes that have been given to them in one way or another. We'll talk about that too. And so therefore, they're not susceptible to this particular antibiotic, and therefore there is no zone of inhibition that's being shown in some of these areas because those bacteria are not afraid to grow right around it because they're resistant to it. So some facts about antibiotic resistance, really it's just the process by which bacteria has become resistant to antibiotics. And this is really a very important thing to talk about here because a lot of students, incorrectly think that we can become resistant to antibiotics as humans, and that's not correct. Humans can have allergic reactions to antibiotics, sure, but that doesn't mean that you're resistant to them, okay? We do not become resistant to an antibiotic. Only bacteria can become resistant to an antibiotic, but allergic reactions, that's something completely different, okay? So yes, you can be allergic and have a reaction with them, okay, with certain ones. I, for example, am actually somewhat allergic to penicillin, believe it or not, but not to other ones. But we as humans are not going to ever become resistant to antibiotics. That is just reserved for bacteria that can become resistant. So that's really important that you understand that so you can understand this whole process in detail. Okay, so please keep that in mind. Now, how does this work that these bacteria can all of a sudden start becoming resistant to bacteria where perhaps they weren't even a year earlier? Because you're going to see soon how quick this happens. Bacteria, for those of you who remember prokaryotes a little bit, are very unusual. They can do some pretty cool things to share genes that we as humans cannot. And you also need to start thinking about here how they reproduce, which is by asexual reproduction. Okay, it's called specifically binary fission, whereas humans do sexual reproduction. That entire process is very different than asexual reproduction, right? They can have lots of offspring very, very fast. They have very quick generation times. And a couple of ways that bacteria can get genes, or actually get genes to some other type of bacteria, those are called transduction, transformation, and conjugation. And so transformation, real quick, is just in general, when bacteria can pick up genes from something called plasmids, but they often get these plasmids from the environment. So just by being in the environment, sometimes they can pick up these new resistance genes, which is a little bit frightening, right? Another way they can do this is by a process called transduction. And this is something I thought I would just mention quickly. This guy over here is a little T4 bacteriophage. And so he is like the stuff in the nightmares of E. coli bacteria because he can infect them. And sometimes when that process happens, he can actually end up giving them genes that end up becoming part of their own genome. And who knows, may even end up conferring some kind of antibiotic resistance. So that's another way bacteria can gain genes, is from viruses, and that's called transduction. And real quick, the third way is called conjugation, and that's actually when bacteria confer genes to other bacteria. It's literally like the equivalent of you giving someone a handshake, and in that process of giving them a handshake, give them some genes as well. So it's like just giving someone some genes. Bacteria can literally do that by what's called the sex pilus, and that is the process of bacterial sex, and it's called conjugation. So if you're more interested in that, you can, of course, take some time and look that up on your own. So bacteria function in those three ways to be able to pick up genes from the environment or get genes from each other, which is, of course, very different than how other organisms operate. Okay, so keep that in mind, as they are prokaryotes, and of course, they can multiply extremely quickly, as you guys already know. All right, so this slide, actually this figure here, shows pretty much how quickly resistant strains of pathogenic bacteria can arise in populations. So kind of keep that in mind. Even at the bottom, the tigecycline that was introduced in 2005, I was looking at some new articles recently about how widespread resistance in some cases is already developed for this. So if you look up here, like methicillin, you can see it was introduced in 1960. We already had resistance in 61. So very, very quick resistance was popping up here, which again is somewhat frightening to keep in mind. But again, I just wanted to mention that so you can see how quickly these bacteria do become resistant to the antibiotics we put out there. So in most cases, it is very, very rapid. All right, something else to talk about is different antibiotic classes. In other words, like I mentioned before, a lot of times with antibiotics, we can synthesize them, in other words, make them based on their cellular targets or the components that we want to target. And so something like penicillins and cephalosporins have been made to prevent the bacteria from actually putting together or synthesizing their own cell walls in different biochemical manners. So it might not allow them to make a certain molecular building block that allows them to put together that whole cell wall. And that is what those particular antibiotic classes target. So in other words, if you take that, okay, and the bacteria in your body are not resistant to it, let's say, then those bacteria cells would die because their cell walls are going to get degraded or broken down. They won't be able to make them, but it's not going to affect your cells because you should be shouting out right now, of course, do not have cell walls, right? Something else to think about is down here. We have the sulfonamides, okay, and then the fluoroquinolones. Those inhibit DNA replication at the bottom, whereas the sulfonamides basically inhibit what's called folate synthesis. Folate, guys, is a metabolite that's used, it's also called folic acid or vitamin B, to be basically in bacteria too, somewhat similar in us, like a precursor in creating, I believe, DNA and other nucleic acids, as well as some amino acids. So in other words, that's really a metabolic precursor they need to make those building blocks. Then over here on the right, we have some other different antibiotics like tetracyclines. Those are pretty common. A lot of you guys have probably heard of those before, if not, been recommended one by a doctor. And then like macrolides, for example, on the bottom, like Zithromax, guaranteed you probably heard of that too. Many of these work by inhibiting protein synthesis. So they stop the bacteria from making their appropriate proteins they need for their cellular processes. So these are different ways of course, targeting bacteria. There's even more that I'll mention later. But the issue here is that, a lot of these pathogenic bacteria are now evolving to be resistant to a variety of cellular targets, which again is pretty scary. Okay, so as far as usage goes for antibiotics here, this is going to kind of help explain to us just some simple medical procedures that would be considered life-threatening if we didn't have antibiotics. So you can see over on the top there, we have transplants, right? And then something like dialysis, which if you remember what that is, we discussed that earlier in our cellular units. It's a procedure usually used to remove waste products and excess fluid from the blood when the kidneys, of course, aren't working properly. And so that wouldn't be possible. Dialysis wouldn't, right? Then down at the bottom there, we have other things like on the right side, hip and knee replacements, right? And of course, just, immune suppressing sicknesses, even including things like cancer. So all of these, right, anything that any human needs, right, is all going to be based on the fact that these are all, processes that require antibiotics so that the immune system can continue to function at its best capacity and these people can survive. So we need to make sure that any kind of, surgery, right, or a transplant or a newborn that needs antibiotics, because it could be susceptible to so many things out there. It's so little, right? We need to make sure that we continue to produce these antibiotics or try to produce new ones if needed, so that these processes are all, continuously going to be possible. So again, they would be considered life-threatening if we didn't have antibiotics today. So kind of keep that in mind, please. Let's see, next we have overuse, right? And so overuse or overprescribing, you've probably heard of before, is something that might happen. And it's really a major factor contributing to antibiotic resistance, because the issue is that sometimes humans, and this isn't happening as much now as it was when this problem was, first announced, humans are being prescribed antibiotics by doctors for things that weren't really bacterial related at all, such as a virus. If you have a cold virus or you have the flu, unless you have a secondary infection threatening you, you shouldn't be getting antibiotics, because those are not going to help work to kill a virus, right? These are designed to kill bacteria cells, not to kill viruses. So overprescription was a big problem. I know when I was a kid, my mom used to say that, she'd take me in and my pediatrician was just like, oh yeah, we'll give her some antibiotics for this or that. And it was usually just viral, for sure, like a bad cold or something. And so there was no need for me to get those, but I did. And so again, definitely not a good thing, because we're putting that out there in the environment. And as I'll explain later, what that's doing, that's selecting for those resistant forms to actually produce more of themselves. The other issue here is that, we can put some percentages up. So about 30% is estimated to be just overprescription. Like I mentioned before, antibiotic use in humans, it's, not something that should be continued, of course, but animals are another issue, and just the continuous use in livestock feed. And now, of course, you've probably seen more and more in packaging, their consumers are picking up things that say that, they're not feeding these antibiotics to livestock anymore. And so hopefully that's the case in some of what we're purchasing. But of course, it's definitely still something we see in a lot of animal feed, which is causing problems, because again, we're putting it out there, right? We're allowing it to be out there. And so antibiotics, again, are like this picture here, supposed to be, just created to fit like a key and only destroying, certain pathogens. But these antibiotics, again, as a means of preventing disease, is really not how they were intended to be used in the first place. And so this is just a major factor causing more and contributing to more resistance. So how does all of this arise? So this is really just an animation that's going to show you some bacteria cells. And if we go through some rounds of antibiotics, this is similar to what you're going to be doing in your lab, by the way. Dose 1 of antibiotics shows us getting rid of course, many of these susceptible bacteria, right? Those are dying off. But then we see 1 go to green, meaning it's picked up a mutation, perhaps, from the environment in your body, perhaps a random mutation occurred. And then remember, these bacteria are going to go through asexual reproduction or binary fission. And so they're going to multiply. And those that are resistant are going to multiply more of their own kind. So by the time we get over here to the second dose of antibiotics, right, we're going to start to see more and more of those resistant forms surviving. So in essence, that's why you should, of course, be finishing all of your antibiotics, even if you are starting to feel better, because in that case, they are designed to hopefully kill off all of the bacteria, including those that are resistant. But the problem is often that people don't do that, right? You start feeling great after a couple of days that you're on them, when you really needed them, of course, for, let's say, strep throat or something, right? And so if you don't take them all, you're actually selecting for the strongest ones to survive, and those that survive will reproduce more of their own kind, which again is creating all kinds of problems, okay? Especially because you're A, throwing out those antibiotics, putting them back into the environment, and B, possibly gonna cause a relapse in yourself, right? Because now you have bacteria in your own body that are resistant to these antibiotics. So keep in mind that this is really an evolutionary process, guys. All right, some of the things you've already learned about make this a perfect case study for us in evolution, because really this entire antibiotic resistance crisis depends on two things, okay? Those two things are evolutionary mechanisms, one being mutation and two being natural selection. All right, I listed gene flow and genetic drift here for you as well, because if you're in my 138 class, you're going to be learning about these as other patterns for how evolution can take place. Now, genetic drift, as we'll talk about, is not going to be necessarily adaptive evolution at all, nor will gene flow, because that's really based on migration. New members, for example, in gene flow can come in or leave populations, bringing with alleles or leaving alleles behind. But in that case, those are not really going to contribute very much to this process. So we're not going to talk about those as patterns of this kind of evolution. Merely, we're going to be interested in mutation and natural selection here. And just to talk a little bit about the bottom picture as well, keeping in mind that's called rifampicin, and it is pictured. This guy can actually destroy the bacteria's RNA polymerase. But what's interesting about this is that only a single base pair mutation, remember your point mutations back in our DNA unit, all we need here is that single base mutation in the gene to actually confer resistance for the bacteria to this antibiotic. So in other words, one base pair can be enough to actually confer that resistance. So again, somewhat of a frightening observation there. And so finally, keep in mind that this crisis is really due to two things, right? An increase in resistant organisms, as we've mentioned, and a limited number of new antimicrobial drugs that are coming about. So that really equals a very problematic scenario for us. And so really the combination of the resistance and lack of new antibiotics is starting to draw the world's attention here to dealing with this big antibiotic resistance crisis, okay? And one reason you might be wondering, why haven't we been producing that many antibiotics, right? Why is that not happening very often? And I'll talk about some hope here in a minute, but really the reason for that is that new antibiotics, even a few that have come out in the past few years, the problem with them is that pharmaceutical companies are actually making more of a profit off the old ones, to be honest. And so because they're less effective, that actually means people are going to be buying more of them. So these companies are making a profit of them. And so if you're a person or a scientist who's working on, new ones, it actually ends up being pretty difficult to get new antibiotics, because you end up spending more money trying to get all those, new antibiotics together than you do in the end when you're making them. So in other words, it's kind of just an endeavor in losing money. So it's kind of a sad thing, right? And then just to discuss the other scenario here in a post-antibiotic era, what would happen if we didn't have them anymore? Just kind of some sad stuff before I talk about the hopeful stuff. Currently, we have 80% of all gonorrheal infections, right, that's an STD, that are now resistant to antibiotics. Again, somewhat scary. Also scary is that about 440,000 new cases of drug-resistant tuberculosis are annually found. So tuberculosis, again, is another disease I'll talk about at the end. There is another type of new antibiotic that's been created that supposedly is going to be helpful against that issue, but we still have some very current scary problems here. In the future, hopefully this will not happen, but in the future, if we don't start figuring out how to solve this problem, strep throat or a scraped knee could end up killing a person, right? Even a simple hip replacement could result in death. And of course, the cost to treat drug-resistant cases is estimated to be at least double. So pretty serious stuff. All right, so please keep in mind that this really should be added to our list of national emergencies. We should be thinking about better ways to make new antibiotics. And so one really neat thing I wanted to talk about today is just something called the iChip, and a new way of really making and discovering antibiotics. That's teixobactin, by the way. It's this new class of antibiotics that they're looking into, and this discovery was made using this really cool device called the iChip. The iChip is really a device composed of almost like hundreds to thousands of tiny little, you can think of them as petri dishes, really, that we can take this entire device and place it in the soil. And of course, why in the soil? Hopefully you're shouting out, but that's where a lot of bacteria are found. Special bacteria that wouldn't normally grow in a petri dish can grow in this thing. And so I'm going to post a video later. You'll see how neat this is, that if you go ahead and plant it in soil, it can be in there for quite a while. You can allow bacteria to grow on it and then take it out once the bacteria have grown on it and do some of your petri dish testing that we discussed earlier and look at some different antibiotics with respect to these bacteria that you're growing on the iChip. And so using this new method, this new technology, teixobactin, was actually a new class of antibiotics found. It actually kills gram-positive bacteria, and it harms them by binding to these lipid precursors that are parts of their normal cell wall. And so far, this has shown a lot of promise. And so it's kind of like our big hope here, is that this new class, maybe because in preliminary studies it showed success and it didn't show a lot of signs of resistance, maybe if we continue to look at new ideas like this, we might be able to find some antibiotics that have more promise, and that are a little bit slower in gathering resistance from these types of bacteria. So I hope all this is somewhat interesting to you. I will post a link later on that talks a little bit more about the iChip and some other mechanisms we're trying to use to design new antibiotics. On the last slide here. Oh, and this of course is something that they are using to kill staph and tuberculosis, which is exciting. Sorry, I forgot to mention that with the gram-positive stuff. Lastly, this is going to lead us right into your little mini lab or your simulation, which you will watch if you don't want to do this at home, but I recommend it, especially because it's very tasty too at the end. My family did this with me. They didn't really have a choice, of course, but we had fun. They got some M&Ms and they mixed them with peppermint marshmallows. And so those were pretty tasty that they got to eat those at the end. But your susceptible bacteria are going to be represented by your marshmallows. You should think about why that is, right? Those are the ones targeted by the antibiotic. The antibiotic, of course, when you read the lab will be the toothpicks. And then your resistant bacteria are on the bottom there of my picture. Those are your M&Ms. Also, you should be thinking about that hard outer shell on that candy and why that would make them resistant, right, to your antibiotic. So I hope if you guys have any questions, you definitely, talk to me, let me know. But we're going to say goodbye to you today. And thank you so much for listening to this talk. I hope you guys are all doing well. And I hope you're finding this a really interesting case study into evolution, natural selection, and how mutations can even give rise to resistant bacteria. All right, so don't forget your bacteria, your prokaryotes. And of course, don't also think that all bacteria are bad guys. They're certainly not. Okay, most of the bacteria in your body are at home there. They're very happy and they're helping you and they're good bacteria. But we're really talking about those that, of course, could end up causing problems because they are pathogenic or disease causing. All right, so again, take care. I will talk to you guys again soon. Thank you so much for listening.